Thank you, Jesper. I'd like to thank Biolamina and especially Jesper for inviting and hosting me here. It's actually my first time in Sweden, a country which I know mainly from binge watching Wallander and Annika Bengtsson on Netflix, and I'm looking forward to seeing the real thing for the next week, hopefully without all the murders, bombings and kidnappings that are familiar from those shows. So I'm going to talk about the area that I've focused on for the last 6 or 7 years, which is induced pluripotent stem cells, and in particular methodology for making them using transient transfection with messenger RNA, which I believe is going to be the method that is likely to prove most applicable as these cells start to get used in regenerative medicine applications. So I'll give you a review of the history and current status of this technology, and got quite a few slides to go through, so I'll try and move as briskly as possible so we have some time for questions at the end. Assume most of you are familiar with induced pluripotent stem cells, but in case you're not, this was a discovery of Shinya Yamanaka, with also contributions from Rudy Yanish and James Thompson in the United States, but he, I think, published the landmark paper in 2006, which showed that it's possible by transiently expressing a minimum of 4 transcription factors that are associated with a pluripotent state, that it's possible to force a somatic cell back to a state very similar to an embryonic stem cell, from which it can potentially be differentiated into any kind of cell, tissue, or organ in the body. And this work was recognized with just 6 years after that publication, with the award of the Nobel Prize here in Stockholm, jointly awarded to Shinya Yamanaka and Sir John Gurdon, whose work 50 years ago on somatic cell nuclear transfer and cloning really established the lability of the nuclear epigenome, and was really the basis for the later work on reprogramming. So why are induced pluripotent stem cells such a big deal? In the near term, I think the applications are mostly are in the research area in terms of understanding the processes by which cells differentiate into different types and subtypes, the ability to make from any genetic background, including a patient-specific background, such as a patient with Parkinson's disease. We can take their cells, we can convert them to pluripotent stem cells, convert those into, for example, A9 neurons in a dish, and study the physiology of Parkinson's disease in that case, and test drugs in a dish. But probably the most exciting thing, certainly I find the most exciting prospect associated with these cells, is the idea that is the potential application in regenerative medicine, tissue engineering. Basically, with these cells, we can take, for example, skin cells from any individual, convert them into pluripotent stem cells, and then potentially produce any kind of cell, tissue, or organ for transplantation back into that person, presumably immunologically compatible. So that's a very exciting prospect. But there is an important hurdle that's been recognized from the beginning, that Yamanaka's original work used viral transduction to introduce the factors that do the cellular reprogramming. And this is using retrovirus or lentivirus, and those factors the so-called Yamanaka factors, those 4 transcription factors, then get integrated into the genome. And those include some known oncogenes. They're all very powerful transcription factors, and they potentially could get reactivated. And it's basically considered a no-no to use cells that have been genetically modified in this way and to produce cells or tissues to go back into a patient. So it's been known for a while that we have to get rid of the viruses. We have to find a different technology to do that. There's been at least half a dozen. In fact, I think there are a couple other methods that are not listed here that have been developed over the last few years. Protein transduction involves delivering the factors as recombinant proteins with cell-penetrating peptide domains. Unfortunately, it's a method which was extremely efficient when it was first demonstrated. It generated a lot of excitement because it was proof of principle, but it is still, as far as I know, a very inefficient and basically impractical method. There are a couple of methods involving excisable monovectors and transposons where one does actually modify the genome to insert the genes for the reprogramming factors, but then one comes back with a transient expression of some kind of recombinase to flip them out and clean up the genome. It's a stopgap solution. 2 methods that have become quite popular, the use of plasmid and particularly episomal DNA to express the factors. So this doesn't have to integrate into the genome. And the use of Sendai virus as a vector, and this is a virus that has a completely RNA-based life cycle. So those 2 methods, they're very easy to use. They've become very popular. I think of them both as stopgap methods because you still have issues with the residual, with getting rid of the residual vector. And in the case of episomal and plasmid DNA, you still have some potential for integration into the genome. And then there's the method of using messenger RNA, transfecting that into the cells to express the Yamanaka proteins, which only has to be done for transiently before the endogenous pluripotency circuit kicks in. It takes about a week to 2 weeks. This is a method which I pioneered and have worked with for the last 6 or 7 years, bringing it to a more and more efficient and simplified version of what we originally developed, and I believe will prove to be long-term the best method for producing iPSCs that can be used in regenerative medicine applications. So this work, I did the original work while a postdoc at Derek Rossi's lab at the Immune Disease Institute, Harvard Medical School. We published it in Cell Stem Cell in 2010. And it created quite a bit of attention. I'll go over the basics of how we were able to demonstrate or reprogramming with messenger RNA in this work. So actually making messenger RNA is quite easy. It uses the same basic technology as has been used for decades to make long single-stranded RNA, in vitro transcription or runoff transcription. You have to add a couple of extra features, 5' cap and a poly-A tail to the transcripts, because once the transcripts are in the cytoplasm, these are required to catalyze or to stimulate the assembly of the ribosome complex and to get efficient translation. If you don't have these features, you will get some translation, but it will be very minimal. Fortunately, there are established techniques for adding both of these features to in vitro transcribed RNA. And if you're interested in that, in the details of that, I do have a publication from 2013 in Current Protocols in Stem Cell Biology, which goes into the pros and cons of the various approaches. But it's basically, it's a solved problem. So how can we get synthetic messenger RNA into cells? The choices are very similar, or basically the same as for delivering any other kind of nucleic acid, such as siRNA, microRNA, or plasmid. and those choices are electroporation, primarily for, which is primarily applicable to suspension cells, or for adherent cells like fibroblasts. You typically will complex the RNA with a cationic vehicle, cationic meaning that it has a positively charged backbone. The RNA itself has a negatively charged phosphodiester backbone. When you mix it and complex it with a lipid or polymer that has these positive charges, that makes it possible, when added to cell culture, that can bring it to the cell because there'll be a surplus positive charge and it will basically just coat the plasma membrane. The plasma membrane is continuously churning and encapsulating and taking up extracellular fluid and large molecules that are outside the plasma membrane through various different kinds of endocytosis. So that leads to the import of the packaged RNA, or packaged nucleic acid, and then with some very low frequency generally, some of this is going to escape into the cytoplasm and become available to express protein. And in fact, in adherent cells, it's quite easy using any of a variety of these lipid or polymer reagents to achieve quite high levels of protein expression using synthetic messenger RNA, probably comparable to what you could get with a well-designed plasmid. However, that expression is, in contrast to what you see with a plasmid, is transient, because the RNA itself turns over in maybe has a half-life of 12, 24 hours if it's well designed, and then the protein itself will decay. If you've got a stable protein like EGFP, for example, you might see from a single transfection into cell culture, you might see several days of robust expression. But for the purposes of reprogramming, we're expressing transcription factors, the Yamanaka factors, MYC, KLF4, OCT4, SOX2 minimally. Those transcription factors, those proteins turn over quite rapidly. And as is shown from data that was generated by one of my colleagues, Wataru Obina in Derek Rossi's lab, the time course for a single transfection, the time course for protein expression, and this is actually looking at protein by antibody staining, is only on the order of a day. So that means if you want to keep up a physiologically relevant level of protein expression, you need to redeliver basically on a daily time course. You need to keep redelivering the messenger RNA to the cells. Now we then run into a major obstacle. When we first tried this, and the first day everything looked great. But you go in the second day and the third day, and the cultures start to deteriorate. You see much less expression, you see apoptosis, you see lack of growth or senescence responses. And we didn't immediately understand what this was. We all learned that double-stranded RNA provokes an innate immune response, but it turns out that single-stranded RNA is pretty immunogenic, and there are a whole host of antiviral defenses that are present in all cells to some degree or another for detecting exogenous nucleic acids that are associated with pathogens. For example, RIG-I is a single-stranded RNA receptor that is universally expressed in cells and activates these pathways. Toll-like receptors 7 and 8 are endosomally localized toll-like receptors that are specific to single-stranded RNA. They're not that widely expressed by default, only found in a few cell types, but when the system becomes activated, they can become induced. Then toll-like receptor 3 is classically thought of as a double-stranded RNA receptor. That's much more widely expressed. And it turns out that it will also pick up single-stranded RNA. And these pathways will activate the type 1 interferons, interferon alpha and interferon beta, and that will lead to the excretion of these interferons, and in a paracrine and autocrine fashion will activate both the cell itself and the surrounding cells and basically put them on alert status and bring the whole, sensitize the whole system. And that has very deleterious impact on your ability to continuously express proteins with exogenous RNA. So this turned out to be a huge stumbling block, and for a while it looked like we weren't going to be able to do it. If you have basic unmodified RNA containing standard nucleotides, this is a panel this is qPCR analysis showing a panel of 6 genes within the innate immune defense network, and we can see that if we have basic unmodified synthetic RNA, we get strong activation of each of these genes, and these genes are involved then in actually recognizing more RNA, so the whole thing is a positive feedback loop, gets worse and worse. Researchers at the University of Pennsylvania, Caitlin Carrico and Drew Weissman, discovered that it's possible to make RNA less visible to the innate immune system by substituting certain modified nucleotides for the standard nucleotides. These modifications tend to occur naturally in eukaryotic RNA, and they're part of the signature or password that says it's okay, I belong here. So basically you can provide that password by incorporating those substitutions into the synthetic RNA. And a combination which works very well is to use 5-methylcytidine instead of cytidine and pseudouridine instead of uridine. This combination together works better than either of them individually and much better than completely unmodified RNA, and we get decent levels of expression. And we found that once we could do that, we could keep up transfection, and the cells would stay healthy, and we'd see continuous good expression for actually weeks on end. So basically indefinitely. We did find there was still some tickling of the innate immune response even when we had these modifications, and through positive feedback through the interferon system, there was a gradual activation after a few days. So we used a interferon inhibitor. Surprisingly, there are not that many molecules known that will block interferon, but vaccinia virus expresses a decoy receptor for interferon called B18R, which is available commercially and blocks this pathway. Incidentally, it is now possible to get around the use of B18R by further purification of the RNA. Turns out that most of the residual immunogenicity of the RNA that's modified comes from double-stranded side products of the in vitro transcription reactions. So we can actually get away without the interferon inhibitor now, but that wasn't true 5 years ago. Once we'd got over those hurdles, it didn't take us very long to get get the whole process working, and what amazed us was how rapid and how efficient the process was. It's actually turned out, normally we expect a so-called non-integrating technique to be very low efficiency compared to the already poor efficiency of viral reprogramming. Viral reprogramming has efficiency of typically about 1 part in 10,000, so 10 to the minus fourth of the cells actually get reprogrammed. But we would see crazy efficiency in some of these experiments, and also the whole process was accelerated down from 30 to 35 days down to 18 to 20 days. You'd see the colonies emerging. And as you can see, depending on some of the specific conditions we use, like a low oxygen condition, greatly promotes the efficiency of the process. You can get crazy levels of iPSC colony formation. The main drawback to the protocol as it was originally published in 2010 was that it was fairly labor intensive. It took 14 to 18 days of transfection, so you have to transfect the cells 14 to 18 days in a row, and they do relapse very quickly if you don't, so you have to be doing that on Saturday, Sunday, whatever. So it was a fairly labor intensive compared to the other one-shot techniques that are out there. and was using feeders, which are feeder cells to support the target cells, which is kind of a no-no for any clinical application, and it also adds to the work involved in the protocol. So when I moved on, I was doing some consulting for a company called Allele Biotechnology in San Diego, and we spent quite a bit of time improving the whole methodology with the aim of streamlining the protocol, eliminating the need for the feeder cell layers, shortening the programming time course, eliminating any need to passage the cells during reprogramming, simplifying the way in which the RNA was delivered. Also, there were, in the original protocol, some animal derived products, which again is something that needs to be eliminated going forward to clinical applications. So we wanted to get rid of those and then have a completely xeno-free protocol. And this work was published in Scientific Reports in 2012. And the biggest key to this was to make the whole thing faster, and the key to making the whole thing faster is to have a more powerful cocktail that pushes the cells through the reprogramming process more quickly. And this was just showing an experiment that we did where we transfected cells from 6 to 11 days using a 5-factor cocktail of four Yamanaka factors plus Lin-28, which we had originally published in the Cell Stem Cell paper. 6-factor cocktail in which we also added NANOG from the Jamie Thompson cocktail, and then equivalent 5 and 6 factors, but using an engineered version of OCT4 that has a MyoD transactivation domain that had been published by Hiroyuki Hirai in 2011 and shown to significantly accelerate the whole process. So each one of these, so independently the NANOG and the so-called M30 engineered OCT4 shave a day off the kind of core reprogramming time, taking it down from maybe 9 days to 8 days to 7 days. And that's key because every day you save reduces a lot of constraints, makes it possible to do the whole thing in one dish. So this is showing, we were again surprised just how efficient and how fast this process could be made using these approaches and using the more advanced cocktails. And here's some imagery, time course imagery of some human fibroblasts, day 3, day 4 of the protocol, day 5, day 6 you're starting to see a bit of formation of sort of foci here, which are the kind of pre-colonies. Day 7, you're already starting to see some colonies appear. And then day 8 and day 9, it's basically chock full of colonies, and we've got efficiency levels here that are maybe 10 or 20%, and compared to 0.01% with lentivirus. So amazingly efficient, and all done under feeder-free conditions and xeno-free conditions, which are prerequisites for clinical application of these cells. So around 2012, I had get a paper presentation, a set of future goals where I wanted to take this to the next level so that we could mass produce these cells, potentially for banking of autologous iPSCs that could potentially be done for everybody on a sort of direct-to-consumer 23andMe type model. Obviously, that's a few years from being a reality or being realistic, but to get it to the point that we'd be ready to go when the demand for that exists. And all of the goals that I set there I've since achieved with the new company that I set up in 2012, Stemiotics, to do this, to bring this to fruition. So conventionally it was taking 4 to 6 months to get iPSCs from a fibroblast line, and costing $10,000 to $20,000 per line. So I thought that reasonable targets based on what I already knew about the protocol were to squeeze the whole thing down into about 4 weeks, and to get the costs of doing it in bulk such that we could offer a service for as little as maybe $1,000 a line. And that is in fact what we're now doing. And you know, practice makes perfect. We've had a lot of practice now. We've concentrated particularly on the stabilization, isolation, stabilization, and expansion of the cells, and improving that process, making it more streamlined. We actually do bulk passaging from the reprogramming wells. We don't pick colonies. We never have to clean them up. Typically, we pick around we passage from the reprogramming plate the whole culture around day 14, and another 2 weeks later we have maybe a P3 culture. Usually we can stop at P3 and ship back to the customer. And we've done, and I should mention that laminin 521 has been a key enabling technology in allowing us to streamline this whole process, eliminate picking of cells, eliminate, and we never do any cleanup on these cells, which is kind of we just don't want to be doing those processes when we do this at large scale, when we're making hundreds of thousands of lines 5 or 10 years down the road. So we've got rid of all that, and so far we've done over 140 lines. We just offer a basic characterization with OCT4 TRA160 staining. So we've had a lot of practice with that, and it's going very smoothly. Our success rate has been over 98% with these lines. The lines to be used in a feed-free protocol do need to be fairly low passage. Basically, if it's passage 3 or passage 4, we've never seen it fail. When you get out to passage 6, 7, the yield tends to drop off, but it's usually still okay. Above that, I think you would have some problems. So we don't advertise this as the way to go for the most refractory lines or aged lines, but that's not really the goal here. The goal here is to be ready to produce, mass produce clinically relevant lines for the consumer market ultimately. So what are our future goals at this point? We're currently totally focused on human fibroblasts. The human fibroblasts are fairly easy to get and establish primary cultures, but there are other candidate cell types that may be of interest in terms of being, they can be obtained less invasively. For example, keratinocytes, which can be obtained from a hair follicle, or renal epithelial cells that can be obtained from a urine sample. Obviously, blood is another candidate. So the question is, are we going to long-term, when we get to a mass production of these cells and mass pre-banking of iPSCs, are we going to continue using fibroblasts, or are we going to use some of these other cell types? And I think that's an open question. The part of the pipeline that we do is we take a say P3 or above fibroblast line that the customer ships to us. We reprogram it and we take it out to maybe P3 when it's relatively stable and pure, and we can ship it back to the customer. So we'd have to integrate into this process the processes of harvesting the original cells from the patient by biopsy or whatever the technique turns out to be, depends on the cell type and the primary culture that's required to prepare the cells for entry into the reprogramming pipeline. And then we'll have to obviously upgrade the whole production pipeline to GMP standards before this could be used to pre-bank cells for clinical applications. But those I think those are the key issues, the technical issues of the reprogramming. It's already at a very, very high level now. And I'll just close with acknowledgements. This is my crew back in San Diego at Semiotics, Hanazai, and Masaru Nidari, and of course many academics and many folks in academia and industry who I've worked with over the last several years to get this to the place that it is today. And with that, if we have time for questions, be happy to take them.
Great, thank you, Luigi.